Okay, now we're set. Okay, so we're going to dig deeper into DNA. So the basic structure of DNA is it's a long linear molecule, and the backbone is a phosphate connected to a sugar. The particular sugar is deoxyribose. That's where it gets its name. It's actually missing an oxygen there. It's a deoxy, missing an oxygen, 5-carbon sugar. Phosphate sugar, phosphate sugar, phosphate sugar. And this goes on for thousands, tens of thousands of repetitions in a typical DNA molecule. And attached to the sugar is what we call a nitrogenous base. So you notice it has nitrogens in it. And nitrogenous bases come in 4 flavors. Some have 2 rings, adenine and guanine, A and G, and some have 1 ring. This is thymine and cytosine here. And this long linear molecule with its sugar phosphate backbone and the bases sticking out to the side can pair up with another DNA molecule that's running in the opposite direction. And the pairing occurs between the bases. They bind to each other weakly, but you get 2 bonds that form between A's and T's, and 3 bonds that form between C's and G's. All of these molecular bonds in here are very strong covalent bonds, very hard to separate. But the bonds that bind this strand to this strand, these bonds here are very weak bonds. And so I've said in a way it's a Velcro-like structure. Any one link is very weak and it's easy to pull apart. But if you have dozens of them or thousands of them, the 2 strands will stick together quite tightly. Okay, let me advance here. So if we drew an atomic model of this, this is the same structure. It's just showing the nitrogenous bases with the nitrogens in blue and the carbons in black. The phosphates here, the sugar is behind. It has the advantage of showing us the true 3-dimensional arrangement of the molecules, but in some ways it's harder to see because you can't see this sugar ring in this 3 dimensions. All right, and the DNA can be represented in this way also. So a good cartoon structure shows the sugar phosphate backbone with the nitrogenous bases A, C, G, and T hanging out on the side, and that pairs up with another long linear DNA molecule. Okay, and in fact, it's not flat like this. It forms the famous double helix. And if we actually build a model in which we represent every atom to their correct size, then it looks like the molecule on the right here, DNA double helix. And I emphasize the fact that even though this looks very uniform to us, if it's a TT base pair, the lumps and bumps on the outside of the helix are different than if it's a GC base pair. And so the different lumps and bumps along the molecule are what are recognized by proteins and what are the way that the code is read is really, it's almost, you can think of it almost as a molecular braille, that the proteins are able to feel along the top of the DNA helix and feel where the lumps and bumps are and read that as ACGA, AACCAT. Okay. All right. So we can show a DNA molecule, a gene, in this fashion here. So we can be represented by showing all the letters in one of the strands. And what you find is that there is no punctuation, there's no markers to say where one gene starts and another gene begins, or stops and one gene starts. There are, however, areas of the sequence that signify start and stop. There are areas of the sequence that tell the proteins in the cell where to start making a gene. So ATG is always the start of a protein coding region. And there are 3 different codons, TAA is one of them, that signifies stop. And out here in front of the first 3 letters that specify start making a protein here, yeah, are sequences that the proteins can read and say, oh, here's the beginning of the gene. Sometime after this point, there's going to be a gene. And if you need to copy this into RNA, bind it here and start here. Okay. Now, the genetic code we talked about briefly, 3 letters make a word, where a word is to specify an amino acid. So we have the example of ATG means start, and ATG also means methionine. You're going to make a protein at this spot, add a methionine. And then there are 3 different 3-letter words that signify stop. So the DNA, the coding region of a DNA, will be a long series of nucleotides, and it'll say start with a methionine, then it'll have CGT, for example. That specifies the amino acid arginine. TGC specifies the amino acid cysteine. And the core sequence of events is that the DNA gets copied into an RNA, which is identical except it uses a slight variant of the thymidine residue called uracil. And then this goes to the ribosome, and it's kind of fed into the ribosome, which reads the codes and assembles the string of amino acids. Now, kind of one last point in this review here is that this is called a codon usage table. So it shows you the meaning, if you will, of all 64 possible combinations of ACG and T, or ACG and U. This is RNA. Now, one point to make from this is that we say that the code is redundant. That is, it specifies 20 amino acids, but there are 64 possibilities. Now, 3 of them are stop codons, but that leaves 61 possibilities. And so there are, for some amino acids, look, for example, here at leucine. Leucine is specified by 6 different combinations. So CUU, CUC, CUA, CUG, they all make leucine. Tryptophan over here is specified by only 1. It's the only one that only has a unique codon. So we say that there's redundancy in the genetic code. And this is important to remember when we talk about mutations. So to jump ahead a little bit, what would happen if this C got mutated and became an A? Well, the answer is almost nothing would happen because they both spell leucine. And so you can get mutations that in fact have no bad effect on, on the protein that's made because of the redundancy in the code. That happens a lot. Okay, now let me, before I go into this next part here, and I want to get rid of, I'm seeing something in the top of my screen that you're not seeing. Whoops. Okay. And let me just pause here for questions on this sort of basic core structure. Anybody have a question that they want explained about any of those things?
I do.
Go ahead.
That about redundancy. You know, across history, we've thought lots of things were useless, you know, like tonsils and adenoids and appendix, and we find out not. I'm wondering what the chance is that we'll find out redundancy is not, that there are actually differences.
Okay, well, we do know that's one of the ways in which it is used. So for example, I showed that there are 6 different 3-letter combinations specified leucine, but they're not all equally abundant. And so if in fact, where this comes into play is on the ribosome has to have these transfer RNAs for all of the codons, and it doesn't make equal amounts of all of the transfer RNAs. Some of them are rather rare. And so if you use a rare 3-letter combination from leucine, the protein will actually get made more slowly, because when it's being made on the ribosome will actually have to sit there and wait for one of these rare tRNAs to bang into it. Whereas if it uses one of the abundant ones, it'll be made faster. And we found that in evolution, this abundance of the tRNA for the particular 3-letter code, in fact, is used to regulate the amount of protein. So you're absolutely right. Evolution has made use of that.
I have a question.
Yes.
On the slide where you had what you call the cartoon version of the DNA, on the one that was shown as the helix, the 3' and the 5' ends were the opposite ends of the same strand. But in the one where it was like 2 rails parallel instead of a helix, you had 3' on both ends and 5' on both ends of the other one.
I think that's come up before, and that is definitely a typo. They should definitely be in the opposite ends.
Okay.
Yeah, if you cut a piece of DNA anywhere, one of the strands at the cut end will have a 3'. That refers to the number of the carbon in the 5-carbon ribose. So different faces of that sugar, the ribose sugar, will stick out at the end. The 2 strands definitely run in the opposite direction. Okay, any other questions?
Yeah, I do. When you showed the 3 different ways of making leucine, I'm presuming that like the U and the A, those represent different molecules, right?
Yes.
Okay. I don't understand how you can use different things, different molecules to make leucine.
Okay, so the important thing is this is the DNA and the RNA are not actually used to make the protein. They are simply a set of instructions, a code, if you will. So the ribosome, which is the protein manufacturing machine, will basically look at the 3-letter code and says, the code says, bring in a leucine. And there's a separate molecule that will carry in a leucine and match it up with a 3-letter codon. And then it'll get added to the end of the protein chain. So it's just the information. It's not physically converted or used in any way. Any others? Okay, we'll move on then. All right, so what is the information that's in DNA? Okay, so it contains 3 billion base pairs, 3 billion bits, if you will. And in a way, if that was a computer program, that's not a very big computer program, really, to make a whole human being. It's pretty amazing. So you get 3 billion from your mother and you get 3 billion from your father, basically. And they're organized as 22 sets, 22 different pieces of DNA, 22 chromosomes. 1 set from your mother, 1 set from your father, and then if you're female, you'll get 2 X chromosomes, and if you're a male, you'll get an X and a Y chromosome. Now, in all of these 3 billion base pairs, there's the information for now about 21,000 genes, which is a surprisingly small number to us. You know, when I was in school, we thought that the number would probably be about 150,000. We were all shocked because this is about the same number of genes as it takes to take a fruit fly. It's also not a heck of a lot more genes than is required to make a fungal cell, which is about 10,000. So the genes, in fact, only occupy a small part of the total DNA that we have on each of our cells. So about 2% of all of these nucleotides code for proteins. And these are called the exons, that sort of a shorthand for they are expressed. They show up as providing instruction to make a protein. Now, 25%, about a quarter of our genome, codes for introns. Now, I briefly told you about introns at the beginning, but let's go and take a look at this again. So let's go back to that gene that I was showing you. And I said the gene has a start here and a stop here, and each 3 letters tell the ribosome what amino acid to add next to making a protein. But almost all genes in virtually all the organisms that you can see, from plants to fungi to human beings, have these what we call intervening sequences. And that is, there'll be a stretch in the DNA that we find is not used to make protein. And in fact, we now know that when this whole gene is copied to make an RNA molecule, the RNA molecule will start up here and it will end down here somewhere. And then when it goes on to the ribosome, this part up here is almost like a leader, thinking of putting it into an old film projector, and the ribosome will read along it until it finds the first ATG and says, ah, I start here. But in that RNA that's made, that's going to be sent, before it leaves the nucleus, there is a mechanism to recognize that, oh, this is not actually protein coding, and it will cut all of this out of the RNA and splice this C to connect it to this G. In other words, it cuts this part out and puts just the needed part, the coding part, back together. And we know that there is sequence in here that's used. These introns, intervening sequences, almost always start with GT, and they'll almost end with AG. But there's got to be more information in it too. For example, here's a GT, but that's not recognized as the start of an intron. Here's another one up here. So this is one of the biggest challenges in molecular biology, is how these introns are recognized. It's something very much a focus up at UCSC, and in previous versions of this course in past years, we've had a couple people lecture on this, and we'll get them back again. The other thing is that this gene is from the fungus that I work on, and the introns are rather small, maybe 100, 200, 300 bases. In humans, these intervening sequences can be huge, 10,000 bases long. And yet, inside the nucleus, they're recognized that they don't really belong in the final RNA. They're cut out and they're destroyed immediately. So why they exist is really still very much an unsolved problem of molecular biology. There are some suggestions that maybe it breaks the protein coding sequence into sort of like modules, so that different pieces of protein as coded in the genome maybe could be put together in different orders for the evolution of new proteins. There are few examples that seem to fit that, but for the most part, that doesn't explain it. All right, so bottom line though, is that although we have all of these genes, a lot of the information in the gene is intron sequence, and that gets cut out before that RNA goes out of the nucleus to be read to make a protein. Now, the other thing is that 50% of the DNA is repetitive DNA. Now, there are bunches of different kinds, but you could have, let's see, a sequence that said GGACG, and it will go CGACG, CGACG, CCAGC, just repeat again and again and again for hundreds of times, perhaps thousands of times. And what the function of this repetitive DNA is, we don't understand. It seems to be, in some cases, sort of act like a spacer, just kind of random bits of stuff put in between the actual genes. A little bit we understand. There is a big region in the middle of each chromosome that's repetitive DNA, and it's used to allow the right chromosomes to get together when the chromosomes thicken up, when the cells divide. But for the most part, its actual function is a mystery. It's found in kind of like all large creatures have lots and lots of repetitive DNA, and some creatures have a lot more than we do, 10 times more than humans do. So another mystery. 15% is mobile elements and the remnants of viruses. So there is a class of viruses that is part of their life cycle, make a break in our DNA and insert their own DNA into that break, and it can stay there permanently. There are pieces of DNA that have the sequence information that they can essentially be cut out of the DNA and then go and insert themselves someplace else. They're called jumping genes. Barbara McClintock, famous molecular biologist, one of the first women to win the Science Nobel Prize, not the first. She was the world's expert on these jumping genes. She found them in corn initially, but they're present everywhere, and there's a huge number of them in our DNA. Now, the bottom line of all this is that if only 2% or less actually codes from the proteins, you know, for a long time we thought the rest is sort of like junk DNA. And that's not a completely ridiculous idea, because we know that you can actually like remove big pieces of introns. You can remove whole introns from genes, and it has no obvious physiological effect. The gene still works, the organism still lives, it seems healthy. So the introns, the repetitive DNA, you can do pretty gross manipulations with them and not harm the cell or the organism. But we don't know if there's any subtle regulatory features. Now, something that's a really hot area in molecular biology today is really investigating this problem. And it's been spurred by the fact that our techniques allow us to actually measure how much of this DNA is ever copied into RNA. And we've been very surprised to find that about 80% of it is copied in RNA. Now, a lot of that, once it's made, it's then degraded right away, but not all of it. And in fact, I think we had Karen Carpenter come speak to us. She's a new young assistant professor, and she's found that some of these long RNAs are involved in regulating genes in our immune system. It's been found that some of this junk DNA makes tiny little RNAs that are maybe 20 letters long, and they help decide whether genes get turned on or off. So we're unraveling more and more what is going on with all this extra. But the bottom line for this slide is that we know the genes are important, but we're finding out that DNA has a lot of other structure and function in every cell, and we only partially understand it. One little side note here, this repetitive DNA. So when you get some of these things that will repeat over and over again, that varies a lot from person to person. So you might have some 5-base pair that in you is, you've got 20 copies, I have 17 copies, another person has 47 copies. It is getting the sequence of how many repeats of these repetitive DNA is in an individual person. That's what's used to do this forensic DNA typing. How do they know if they find a DNA sample if it is the match to somebody that has submitted a DNA sample sometime in the past? They're really not sequencing their whole genome. That would still be a huge and rather expensive project. But they can sort of quickly just measure, say, 20 sites for different repeat numbers, and you end up with basically a 20-number code that is unique to every individual. All right. Rather complicated how it's actually done, but one use of the repetitive DNA is turning out to identify criminals by their DNA samples. Okay, so I want to turn to a couple of properties of DNA that are important in understanding both its function in the cell and important in understanding some of the laboratory techniques that have been developed to make us able to really see in the lab the details of DNA. So one important one is this phenomenon of DNA hybridization. Now, what this means is the following. So most of the DNA is present as a double helix, two strands bound to each other by the base pairs. And as I said, in our cells, each of these strands can be tens of thousands of bases long. Now, if we extract the DNA from a cell and we put it in a test tube and we heat it up to about 200 degrees, that's pretty warm, but it's not enough to actually cook the DNA. It'll still stay intact. But what will happen is as it gets this warm, there are all these molecules, it's in a solution in water. All the water molecules are jiggling around, everything is banging around, the whole DNA is vibrating as it gets hotter and hotter, and the two strands will actually come apart. They will rip apart. So the double strand will become single-stranded. All right, because of the high heat. The interactions between the base pairs is not strong enough to stay together at this temperature. Now, then what happens if we then slowly cool it back down again to body temperature, 98 degrees? And what we find is that what will happen is the strands will go back together. They will find each other. Even though you've got tens of thousands of base pairs, they will match up so that once again, you got TTTAA, and that will match up with AAATT. This is the hybridization phenomenon. The more long DNA you have in here, the longer it takes for that to occur. But they're all sort of bouncing around until eventually they've matched up perfectly once again. And you can even test this functionally. You can inject this DNA back into an animal and see if it functions, and it functions. You can take it apart and put it back together. Okay, now here's an important variation and use of this. This occurs in living cells and it occurs in the lab. Let's say we heat up the DNA, we separate the strands, but then we pick a region of a tiny region of the DNA and we synthesize an exact copy of this. This, by the way, practically is easy to do. You can just ask a company to do this for you, type it into the computer, and it arrives FedEx the next morning, costs about $5. So you get a little tube full of these, all identical. I think I've got about 18 letters in this little sequence. And you take some human DNA, you warm it up to 20 degrees, the strands come apart. You have in your test tube also all these little single strands of DNA that you have added. And now what happens when you cool it back down again? And the answer is that these strands are banging around there and they're going to be encountering DNA strands, and they will eventually, where eventually is actually like in 2 or 3 seconds, they will find and bind to the complementary strand. So after you cooled it to 98 degrees, what you will see is in this region, that is the exact complement, we say, to the small piece of DNA we added, we get a small double helical region. This is the sort of magic trick behind so much modern molecular biology, because you can see it allows us to find to essentially tag in any way any gene among these billions of base pairs. We just make a little piece of DNA that has the exact sequence of a piece of that gene and do this DNA hybridization experiment. And the piece that we made will now end up attached to our gene. And we can exploit that in several ways. Now, and I'm going to talk to you about that at the end. Now, this is also the core phenomenon behind the whole CRISPR phenomenon that you've already heard about, gene editing. How can we go in and change one gene among the 21,000 genes we have? Well, we can do that because we can make these small bases. And if we wanted to change this A to a T, for example, we could make one of these and make this one change in here. And CRISPR exploits the fact that when we put it in with a mix of all the cell's DNA, our piece will find its target, bind to it. And then if you add the appropriate enzymes, you can actually turn out inserting this piece precisely into its match. All right, so I'm not giving you the details of that, get rather complicated, but I want you to understand the core thing here is that this amazing phenomenon of DNA hybridization just explains a lot of what happens in living cells and explains a lot of how we're able to manipulate DNA in the lab. Okay. All right, so now I'm going to move on to replication, but any questions on this last bit here, DNA hybridization or whatever?
I have a question.
Yes.
So when that little piece gets attached, what happens to the rest of the strand? Does it get other things get attached, or does it prevent other things from getting attached?
Yeah. Okay. So it's complicated. So there are lots of possible things that can happen. So in the simple experiment that I've shown here, we exploit the fact that this will bind very quickly because we've added so many of them that one of them is going to find its target. And so at some point early on, you'll actually have this situation where there's only this double-stranded part, and the other part is kind of slowly trying to find its mate among the other billions of base pairs. And so enzymes can exploit this situation because they recognize this as an odd region where it's double-stranded in some part and single-stranded in other parts. And one thing that can happen is enzyme can come here and actually cut at this place and allow us to insert things in there. It's a poor answer, but the answer is a lot of different things can happen. All right, any other questions about this? Okay, I'm going to move on. All right. So how is DNA replicated?
What I do, Barry.
Yep. Go ahead.
I don't know if this is the right time to ask the question, but of the article I sent you, now that we know the physical structure through the process, the IA process that was described in the article, is this the right time to say how that is going to aid us tremendously in understanding this?
Now, were you talking about the protein folding?
No, this was the other day I sent you. It was in Science version somewhere, and it indicated that with the AI, they were able to put the physical structure of the molecule together to see it, and that would make it easier to now. Many things will be easier to now that we know what it really looks like.
Oh, okay. Well, I guess the shorthand is that it's a pretty different phenomenon, Constantine.
Okay.
I
all
could
right.
come back to that.
Okay, no problem.
All right, DNA replication. So when Watson and Crick first discovered the structure of DNA. You know, the most famous line in their paper was the last sentence, in which they said, "It has not escaped our attention that the structure of DNA shows a clear way in which the molecule could be faithfully replicated." So what they were saying is if our genes are made up of 2 strands of information, then you can just pull the 2 strands apart, and each strand provides the exact information for making a replicate. So if you just copy this strand, so if that's AAT, and you attach to it a TTA, then you've duplicated the double helix, and you do the exact thing on the other strand also. You just, that one would anyway, you duplicate it. And in the core of that idea is correct. So the basic way you make DNA is that you separate the 2 strands so that the double strand becomes a single strand. And then you have enzymes that can say, this is a T, I'm going to attach an A at this position. Now this is another T, I'm going to attach another A, and just move down there and replicate the whole strand. However, it turns out that it's a little bit more complicated than that. So we just mentioned a couple of the complications. First of all, just picture having any kind of thread in which it's a twisted helical thread. If you pull that apart, it's not as though you get 2 simple strands. What you do is you twist it all up at the places kind of outside of the parts you're pulling apart. So you have to not only pull it apart physically, but you have to keep all this twisting from happening. And so there's a whole elaborate system called a helicase, which is basically an unwinding enzyme that not only pulls it apart, but takes out all the twists. Right. I'm not going to go into the details of that. The second thing is that it turns out that there are no enzymes that will simply bind to a naked strand of DNA and add the first nucleotide. You think of there's a problem. I mean, here's a blank one here. If you wanted to start making DNA, you can't, and this is a T, you can't add an A there because those bonds are so weak. They're not actually covalently attached. What you've got to do is to add to the end of a previously existing double helix. And DNA can't do that. So what has evolved is you always start by making a little piece of RNA. So the copying enzyme can do that sort of trick. So you make what we call a primer because it primes the synthesis. You have this naked strand, you add a little piece of RNA, and then you want to add to the end of this. You say, this is an A, I'm going to add a T right here, and I'm going to attach my sugar phosphate bond. Then I'll go and I'll add the next one. Then there's a third complication, and that is that these 2 ends look different. This is the 3', this would be the 5' here. This would be the 5', this would be the 3'. They're chemically different shapes. And so you would have to have whole 2 sets of enzymes, one that could synthesize forward, one could synthesize backwards. And the cell's got to deal with that. All right. Now, the way this all occurs, I think, is best explained. It's all done with proteins, with enzymes. I found years ago these really nice, simple animations. And I'm going to show you this to illustrate how DNA is actually made. So the simple one is the first strand that's made in the right direction. So the unwinding enzymes comes along and it opens it up. Now, DNA hybridization makes this want to snap back together, but there are a set of proteins called single strand binding proteins that prevent them from rejoining. They essentially hold the strands apart, keep them as single strands. Then the enzyme comes in that can make a little RNA piece that matches the base pairs on the DNA. This is called your RNA primer. And now you've got a little double helical region here. Then the DNA making enzyme, DNA polymerase, it synthesizes the DNA polymer. It can start on that RNA primer and keep adding bases. And it always adds to the 3' end. And as this continues to be unwound, it continues to make one long DNA strand. So that's pretty straightforward. And that's the way we thought that we would explain the whole thing. But you've got to do something about this RNA. But you have the problem that the other strand is going in the wrong direction. It's going in the opposite direction. So that's called the lagging strand. So how do you synthesize the lagging strand? Well, basically the same thing happens, is that you add a little RNA primer. And then once you've got the primer in, so here we add our primer, then the DNA polymerase will come in and it'll start adding to the end. The strand runs in the opposite direction, so synthesis goes in the opposite direction. Then you make another RNA primer. So as more and more of this is exposed, every so often you add an RNA primer. But the consequence is the DNA is made in pieces. And they're called Okazaki fragments, named after the Japanese professor, Professor Okazaki, who first discovered this. Now, the way the cell deals with this is that it removes those RNAs and it fills in the gaps. So here we go. We start out on the lagging strand. We make the primer. Now we've got something to add to, so the DNA synthesizing enzyme comes in and it keeps adding to the 3' end. Then you come down here and you make another RNA primer. All right. And after that's done, the DNA enzyme will come in and it will make the DNA until it bumps into that little piece of RNA down there. Then a third enzyme comes in that does a double job. It will come in and it will remove the RNA. Here it comes in, and replace the RNA with DNA. That still leaves a little gap, and there is a final enzyme called the ligase, ties them together, that fills in the little gap. And so that's the synthesis on the lagging strand. So here we have then the whole enchilada, if you will. So we see that as the DNA is being unwound, the leading strand, once it's gotten started, can just continuously make one long copy. But the lagging strand, the whole thing is more complicated because it continually has to add primers and then make a piece of DNA and then remove the primers and fill in the gap. So it's all pretty complicated. But in fact, it's a lot more complicated than this. Because of course, if you really do it in 3D, I mean, how do they access the leading strand and the lagging strand? And it's been worked out over the years, so that I'm going to show you now another movie, which is an attempt to show the actual molecules in their real size at the
DNA replication,
at the speed at which it
helix
actually occurs. And it is an amazing process. So what you're seeing here, the big loop, this is actually the lagging strand. So we'll make a big piece of it, and then it will finish that. So here's the unwinding enzyme. Here's an enzyme that's involved in kind of dealing with this big loop that forms. It all goes at a rate of, synthesizes at a rate of several hundred per second. All right, you've got to copy 3 billion base pairs. It's all done in pieces. I love I'm not playing the sound of this movie, but the last part of the movie said, And it should be noted that although the process seen here is quite complicated, we're only showing a small part of what actually occurs. All right. So this is kind of one of the wonders of things that happen in the cell, is this amazing mechanism that is involved in the synthesis of DNA. All right. So let me mention one last thing about this, and that is that mistakes happen. They can be recognized and they can be repaired. So the DNA polymerase, the enzyme that actually keeps adding the DNA nucleotides, has in it what is called a proofreading function. So you can see that this enzyme, for example, so after it had added this A to the growing strand, it came up to the next one and said, there's a C there. That means the next one over here should be a G, so it adds a G. But then here it made a mistake. It put in a T instead of a G. And now it's ready. Oh, whoops, let me go back here. Now it's ready to do the next one. It's going to add an A here. But on the surface of the enzyme, it knows from the shape of this GT pair that it's a misfit. So the enzyme can kind of feel the shape of this and say, that is not a correct base pair. It's not ATTAGCCG. And what it does is it actually will cut this off right here. It will remove the T and try again, and we'll put in the C, and then it will move on. And there's always a trade-off. Here in the replication of DNA, you find the same thing in making protein, between accuracy and speed. It's just a property of life. So the more accurate you try to make it, the slower you have to go. The faster you go, the more mistakes you make. And interestingly, in the lab, you can buy these enzymes if you want to make DNA in the lab, important applications. And there are some that are fast but inaccurate, and others that are slow but highly accurate. And depending on what you want to do, you'll get one of the others. All right, let me allow for any questions here on DNA replications, and then I want to say more about mutations.
I have a question.
Go ahead.
These Ozaki effects.
Are you muted?
Oh.
Okay. Oh, I can't hear you. Oh, no, say it again. It was me.
There we go. All right. Can you hear me now?
Yes, yes.
Okay. These Ozaki fragments, are they replications of what's already there, or does it somehow know what it's supposed? Where do they come from?
Well, so as you saw on the lagging strand, when it's first made, it's made in fragments. But then those fragments actually get connected by enzymes. So the RNA pieces are removed and they are connected. So they only persist for a short period of time. They were actually hard to find because you can see the DNA is making it so fast. There are only really a couple, but the fragmentation is just an intermediate step in making the whole strand. Does that make sense?
Kind of.
Yeah.
This is hard. And the other thing is, you were just talking about the speed of replication. Is this, would you also think about this in terms of animals with, say, very short lives compared to animals with longer lives? An animal with a longer life can take more time, or is this just relative to heat and cold and when you're tampering in the lab?
Well, the one part that I didn't show you is that when you want to replicate a piece of DNA in a human cell or any animal, you don't really just start at one end of the molecule and move all the way through. Even if it goes at hundreds per second, that would take a long time to make that DNA. And so, in fact, what happens is along the whole strand, there are places where it's opened up. You get like bubbles in the DNA. And so it's replicated in 100 stretches all along the DNA. And then the bubbles kind of converge so that you get one continuous piece in the end. And I'm doing a little hand waving there, but it's actually kind of understood how the bubbles actually go together in the end.
Yes, I have a question. Can you hear me?
Yes.
With the separating of the strands and the changes that you just showed, am I correct in thinking that not only is this done this is done both in laboratory and nature in itself does it within our body?
So what you saw there in the animation was kind of how we think happens in the cell. In the laboratory, it's really difficult to reconstruct all of the whole regulatory system that makes this go smoothly and air-free. So in the lab, we still do it very crudely, but in your bodies, there are probably about 50 different kinds of enzymes all involved in unwinding, unraveling, duplicating, proofreading. It is an amazingly complex and yet highly efficient process.
So what you showed us was what really goes on in the body.
Yes, that's right. That's right. Okay, others. All right, so let me talk just a bit here about mutations. And this is something that, comes up in everyday life. It's kind of important to understand. So I'm emphasizing the point here that the cell really goes to great lengths to make sure that mutations don't occur. But if you're making 3 billion base pairs every time the DNA gets replicated, and actually you've got to do mothers and fathers, so 6 billion, mistakes will happen in the end. And we find that the mutations that occur in cells, about 2/3 of them, 65%, are due to replication errors. The error rate is much less than 1 in a million, but 1 in a million just adds up if you've got 6 billion. Only about 1/3 of the mutations are due to some sort of damage in some ways. And some of those are things that are just inevitable within the cell, and some of them are external. This all gets to the question of, if Uncle George got cancer, was it because of the way he lived, or was it just his bad luck? And the answer is, most likely, it was just bad luck. So the third of the mutations that are not replication errors are, first of all, you can get chemical or environmental damage. And one way this happens, for example, is oxidation damage. That is, oxygen, and we absolutely have to have oxygen in our bodies, is a very reactive molecule. And some of the metabolic processes in our body generate peroxide. We have enzymes to degrade this rapidly, but we don't get 100% of it. And so some of the DNA damage is caused by the reactive oxygen that we just generate, an inevitable consequence of the complex chemistry that's going on in every cell. Ionizing radiation can cause mutations in DNA. Now, some of this is just unavoidable. There are cosmic rays zapping us right now. We are all pretty radioactive. You've heard that there's radioactive carbon in your body, carbon-14, but there's even more radioactive potassium. This is the major salt in your body. We're all about more than 100 millimolar salt, potassium, and a small part of that is radioactive. The measurements have shown that if you you concentrated all the radioactive molecules in your body, this is just from the natural environment, and held it up to a Geiger counter, it'd be about 40,000 disintegrations per second. Really a lot. Then, of course, there's sunlight. And then there are real external things, chemicals in our food and environment. Many of these chemicals being natural products made by plants to avoid being eaten. So cells have many enzymes to repair DNA, many different types of enzymes. So they can recognize, if you had up here an AA base pair, that would be seen as an abnormal lump on this piece of DNA. They have enzymes that would go and cut that out and repair it. If your DNA breaks, and that happens, they have enzymes that will put the ends back together and repair it. We have many different types that are involved in repairing the DNA. And having said this, that all these mutations are occurring all the time, it's important to remember to not worry about this too much because very few mutations are harmful. And why is that so? Well, first of all, as I said, 98% of your DNA appears to not code for any proteins. And mutations in that 98% rarely seem to affect any function. So if you got a mutation in an intron, for example, it is highly unlikely to have any effect whatsoever on the function of that gene. The other thing is that, if it's in the coding region, you might change one 3-letter code for leucine to another 3-letter code for leucine. So it won't have any effect on the gene. The other thing that's really important is that what if a cell in your liver gets mutated and causes a change that knocks out some absolutely essential enzyme? Well, that cell will die, but it will be recycled and replaced. It's just one of trillions of cells, and so it won't really have any long-term consequence. The most important mutations are ones that occur in important genes in the cells that make egg and sperm in your germline. Those are the mutations that have the biggest effects. And of course, mutations can occur that can cause cells to start to grow in an uncontrolled fashion, and that's what gives rise to tumors. All right, but lastly, it's not necessarily a bad thing. Mutations are what drive evolution. It's how we got here. It's just these changes are occurring all the time, and we're doing the test on whether they increase our fitness, in which case you might produce more progeny, or they decrease our fitness, in which case those mutations will be eliminated from the population. And the thing to know is that even in the modern world, where we feel we can control everything, this rate of mutations has not slowed down, probably accelerated, actually, because we have a somewhat higher level of mutagen causing chemicals in our environment, although it's probably not a huge difference. But we are changing as a population at the same rate that we have for the last many hundreds of thousands of years. Okay, any questions on mutations there?
Well, I do have another question.
Yes.
Proteins, as I have understood it during these courses, proteins in your cells tend to be the harmful things. But do they have any relationship to proteins in food that we eat? Are those two separate types of proteins?
No, not at all, not at all. So for example, one of the important proteins in the world is the protein that does photosynthesis in plants. It's a very abundant protein. When you eat spinach or cauliflower, for you, what was an important photosynthetic machine is now just food. It's just a protein food. I mean, so when you eat cows, you eat beef, the protein you are eating was functional protein in that cow. Most of it was involved in the function of muscles, etc. So no, one organism's kind of essential machinery that runs the cell is just another animal, another creature's food.
Well, I understood that many of the damages or problems within our cells were caused by harmful proteins. Have I got that wrong?
I'm not sure I quite understand what you mean by harmful proteins. Can you ask your question again?
Well, I had understood that some of the damages and problems in cells was caused by harmful proteins. Maybe I have got that wrong somewhere.
Yeah, okay, it's a little, I think we'd have to talk it through a little bit more, but I think mistakes in DNA can have the effect of making proteins non-functional, and that can end up killing a cell because it loses some chemical reaction that it needs to survive. But I think in terms of actually seeing proteins as harmful proteins, there are a few rare examples, but that's not a general case.
Oh, thank you.
Okay. All right. So let me go here. All right. So what I want to do today, and I'm going to keep you right till 12 o'clock because I want to do something a little different now. And that is, I want to show you how do you actually work with DNA. And part of the reason that I want to do this is partly to sort of demystify DNA a little bit. I mean, it is this quite amazing molecule, but in fact, we've worked out in modern molecular biology some amazingly easy and cheap ways in which we can do amazing experiments with DNA in a rather simple way. And I'm going to show you how to do this. So we're going to do a kind of thought experiment. And I'm sitting here in my kitchen, and I'm going to show you how you, in your kitchen, could actually do a meaningful experiment with DNA. So let's start with this hypothesis. So it is very likely that some people are resistant to the coronavirus that's infecting us because they probably have a mutation in the spike protein receptor. Now, here's what I mean by this. So the coronavirus attaches to our cells by this spike protein, and it can only enter cells that have the proper binding protein for it in the human cell membrane. And one of the key ones is a protein just called ACE2. So if you had a mutation in the human protein such that the surface of this receptor was a little different shape, it might not bind the spike protein, and the spike protein would not be able to get into the cell. Now, for other viruses, it's true for HIV too. People with these kinds of mutations have been found. And so what if you wanted to do an experiment that somebody had found a family, let's say, that seemed to be resistant to infection by the coronavirus, and you had access to just some blood samples from the family, and you wanted to find out if this hypothesis is true. What would you need to do? You'd need to isolate DNA from one of these people that had resistance. You would then need to isolate the gene that encodes the ACE2 protein. And then you would need to get the sequence of the gene to see if the mutation was present. Now, this sounds pretty sophisticated, and it is, but I'm here to tell you that with just a little bit of equipment, you could do this at home in about one day. So I'm going to stop sharing here so that you can see me. Okay, you might go into speaker view. Am I in speaker view? So because I want to show you some things, so make me full screen on your side. So how do you really do this with DNA? Here we are in my kitchen. All right, so we need the tools of a molecular biologist. And how is this really done? So first of all, maybe surprised to see that most molecular biology is done in microfuge tubes. These are these little tubes here. And we do most of our experiments in very tiny volumes, in part because we don't need a lot of material, and in part because some of the enzymes that we buy are fairly expensive, so we only want to use tiny amounts. Now, the other thing to be a molecular biologist is you have to have this implement here, the pipette man. It's been named the pipette man for 40 years. Some people call them pipette persons, but it actually says on the side here, pipette man. All right, and this is our dispensing tool. So we have all these little kind of plastic tips that go onto the pipette man, and we can measure volumes accurately with this. So, for example, a typical reaction, I've got this set for about 20 microliters. You see how much this is. Milk shows up really well, I found, so I'll just put some milk. That's 20 microliters volume. Can you, yeah, you can see where the milk filled that. Okay, if I put it here, you can see if I squirt this out, it'll come out as a drop inside of the tube. Now, the other thing that we need, we're going to be molecular biologists in our kitchen, which might be a little complicated, is we need a centrifuge, because we need to separate soluble stuff from insoluble stuff. No problem. I happen to bring a centrifuge home with me from the lab. You can buy these, you can probably get them on eBay for, you know, $20. So a centrifuge, you know, is just something that spins things. So here's the little on button for this one. You see if I turn that, let me put a couple of tubes in here just so you can see them spin. All right. Got to make sure that their balance is one on each side, otherwise it'll be out of balance. And I turn this on and off they go. Okay, so with these basic tools, we could start our experiment. So what would we do? So in step one, find one of my test tubes. We would need some DNA from that person that we think might be resistant to the coronavirus. And a simple way to get it would just be to get a blood sample. So if you had a collaborator, they could FedEx a little bit of blood, and you would take maybe 20 microliters of it, put it in your tube here. And in the tube, you just put a mixture of salts, and you would control how acid, you control the pH of this. You'd exploit the fact that DNA is soluble at a certain level of saltiness and a certain level of acidity. It's soluble in different ways than, say, proteins or some of the carbohydrates. You also want to break open the cells so that the DNA comes out. You want to isolate the DNA from this person's blood cells. I should say that it's going to come mostly from the white blood cells. It turns out red blood cells have no nuclei, so don't use red blood cells. You're using the white blood cells. Now, for human cells, this turns out to be pretty easy, because what happens is this is a fairly dilute solution, and when the cells go in there, there's a lot more water on the outside than there is in the inside of the cell. The water rushes into the cell and they explode, literally. It's called osmotic lysis. The water moves by osmosis. Lysis means to break. So if we put blood cells into a dilute solution, salt, controlled pH, they'll all break. So now we got a mix of broken cells, membranes, coagulated proteins, soluble DNA. And what we would do is then we would take these and we would put them in our little centrifuge here, and we would spin that for a minute or two. And after we've done that, when we pulled it out, we would find that all the solid stuff has gone to the bottom of the tube. And so we could just get in here with our pipette and we could type out the clear solution that's got our DNA in it. Now there's a whole bunch of other stuff in this too. Everything that was in those cells, all the other proteins, all the sugars, the lipids, et cetera. There's a lot of soluble stuff in there, and we want the DNA. Now, in the old days, you know, when Rusty and I were first starting to do this, the next steps would be a little bit more complicated because we would actually have to get, we would precipitate out the DNA and wash it, etc. Nowadays, almost everything is done with kits. They have kits to make everything simplified. And so you can buy these kits that make these little columns that allow you to purify DNA really easily. So this comes in a little test tube, you see. And what's here is a little column. And right in there, you can just see that white line. What that is some finely ground glass. It was found years ago that DNA will bind to glass if you grind it up very finely. It will bind to DNA, it will bind to glass if the saltiness and the pH of the solution is just at a certain level. So we've got our soluble DNA, and what we do then is we just pipette it into this. So it would fill up the top part of that. And then we put it into our little microfuge tube here. We put it into our centrifuge and we spin it, all right? And what happens is that the DNA in solution goes through the ground glass and the DNA sticks to the glass, but everything else goes through. So it really concentrates all the DNA in that little bit of ground glass. Now, what we would do in practice then is we then add a wash solution because that ground glass is going to be wet and there's proteins in that wetness, etc. So we'd wash it once or twice. We'd put a solution in there. We'd put it in our centrifuge again and we'd spin it. But now we've got the washed ground glass with the DNA on it. And what we do is we take it and we put it back into our test tube here. Get that top open. And now we just put in pure water. And what will happen is as the water goes through there, there's no salt, the pH is different, the DNA will no longer stick to the glass. And after we spin it, it'll come out and we'll end up with, you can see just a tiny little volume there, typically about 50 microliters. And that should be our DNA, and it should be pretty clean. And the whole thing takes 40 minutes to do that. Now, how do we know how much is in here? And how do we know if it's really clean? Now, that's a little harder to do in your kitchen. You'd have to run up to the lab. All right, we'd have to run up to the lab. And I'll show you how you do that. I'm going to go back to sharing my screen. But this is a rather cool development in the last 10 years of how you really can measure your DNA. First of all, you've only got this tiny kind of double water drop full of DNA. So what you do is you go to the nano drop. There are a number of them around the department. This probably, I don't know, it probably cost about $1,000, you know, not size of a shoebox. And the way this works is it is a spectrophotometer. It exploits the fact that all chemicals will absorb light, but different chemicals absorb different colors of light. And DNA has a very characteristic absorbance of certain wavelengths of ultraviolet light. Now, what you do is you lift up this little lever here, and there will be a little glass plate on it, a glass plate that's only like a couple millimeters thick. And you put on there, you got your precious samples, only got 50 microliters. You put 1 microliter of solution on this little glass plate right here. And let me move back here. And then you lower the lever back down. Get my cursor, and this is a light fiber, and it will shine light through that thin layer of your solution, and it will shine light of different wavelengths. And then this is connected to a computer, and you look on the computer, and it says, here's what your DNA looked like. Your sample did not absorb much of anything out here in the sort of blue region. It absorbed a lot of the light at 260 nanometers. That's sort of short UV light. And then it absorbed less light when we got down to the super short UV. If there were any protein in here, that absorbs light at 280 right here. But we don't see a peak there. We don't see a bump on our curve. We see a nice peak with a maximum at 280. So that tells us, 260 rather, that tells us that this is pretty clean DNA. And furthermore, how high this peak is, how much light is absorbed, tells us exactly how much DNA, what's the quantity. And the machine over here says that the DNA in there was present at a concentration of 1,950 nanograms per microliter of volume. And so in literally 2 minutes, you can analyze the purity and the quantity of the DNA with only a single microliter, 2% of your tiny little sample. Okay. Now, the last part. We've now got the DNA from this person. How do we get the gene? We want to isolate the gene for the ACE2 receptor protein. Out of this 21,000 genes in the DNA. This is the needle in the haystack problem. And this has become feasible, easy even, because of the development of this procedure called PCR, the polymerase chain reaction. You've probably heard or read about this. And I'm going to explain it kind of quickly. Now, let me say, this is kind of like learning to do something new on your computer. Once you've learned it, you say, oh, it's easy actually, but it can take some time to understand how to really do it. And I'm not going to have time to really go through this step by step, but let me give it a try here, how this works. And I've taken this slide from a Khan Academy talk. There's good explanation for this. There's tons of videos if you want to see how PCR works to go over this afterwards. But it exploits the two phenomena we've talked about earlier, and that is DNA hybridization and DNA replication. So we add our DNA that we've purified to a little test tube, and we heat that DNA up so that the two strands come apart. Here's the top strand, here's the bottom strand. Then the trick is we have made two little pieces of DNA that correspond to the end of the gene we're trying to isolate, the ACE2 gene. So we've made a little bit of DNA on the one strand at this end and on the opposite strand at the other end. And the region between these two strands is the gene we're trying to get. And you can do this now because, of course, they sequence the whole human genome. So you can look up where the gene is located. You can get the entire sequence. You can just copy a little set of 19 nucleotides at the two ends. You just order those overnight, cost you $10 to get them. They come the next day and you're ready to do your experiment. So step one, heat the DNA, the strands come apart. Step two, you have your little primers in there, and as you cool down the DNA, the primers will bind to the right part of the DNA. They will bind to your gene. They will find that one needle in a haystack due to DNA hybridization. Step three, you have in your test tube the enzyme that will add to this primer and replicate this whole piece of DNA. So it's going to add to this primer and duplicate the DNA in this strand, add to this primer, duplicate the DNA in this strand. That whole process of heating, cooling, replication takes less than 5 minutes. In fact, it can, in principle, be done faster than that. Then you repeat the whole thing. You heat it up again, and these strands will come apart. These strands will come apart. Your primers will bind to this end of this strand and to this end of this strand. You add the enzyme that replicates the DNA. It'll make a new strand here, a new strand here. And you've now gone from 1 copy of your gene to 4 copies. And then you do it again, and you got 8 copies. And you do it again, you got 16 copies. Typically, you'll do about 25 cycles. And that means you end up with billions of copies of this gene that you want to isolate from the whole mess of 21,000 genes in that test tube. Now, in the end, you can take this and you can put it on a little gel to purify it. The way this works is this is a DNA ladder. It's something that you can buy as a reagent. So this is a piece of DNA that's 200 bases, 300 bases, 400, 600, 8, 1,000, etc. You know how big your gene is that you're trying to get because you could look it up, and you say, oh, the piece of DNA should be about 1,000 bases long. And if this is your 1,000 base pair marker, and here's what came out of your PCR reaction, you say it's exactly the right size. It's almost certainly the right thing. And so now you have isolated the gene from that person who may be resistant to the coronavirus. So last step, we want to find the sequence in this gene. And how do you do that? Well, in the old days, you'd have to sequence it yourself, and it would take a few days to do. Nowadays, what you do is you take 3 microliters, tiny microliters, you put it in a test tube, you ship it off to Berkeley, FedEx overnight, and the next afternoon, the sequence arrives on your computer, 1,000 bases, cost you $4.50. It's amazing. And what you can do then is just use your computer, and these are all publicly available programs, easy to access, and say, I want to compare the sequence I just got with the sequence of this ACE2 gene in 15 other people that I think were susceptible to the virus, and compare it to this person that I think is resistant to the virus. And you might get a result like this. Now, one thing you would find out is that in all of these 15 people, there are actually a number of changes scattered throughout their different sequences. So look over here, for example. We find that this particular nucleotide in the sequence doesn't seem to be too important. In this individual, it's an A, and sometimes it's a C, and other times it's a T. It says that it's at a position that apparently doesn't encode a very important part of the protein in that, a very important amino acid in that protein. But you could say, my prospective resistant person here has 2 different changes in a part that is invariant in everybody else. And this might be a candidate for a change in 1 or 2 amino acids that might make this receptor unable to bind the spike protein, and might be the reason why this person is resistant. So that whole process, interestingly, could take 2 or 3 days to go from getting your DNA, isolating it, getting the gene, and getting this actual sequence for that particular person. So my point is that the technology has become amazing, relatively simple, surprisingly inexpensive for most kinds of experiments. All right, so I ran through that rather quickly, but I wanted to give you some kind of real world feel for what it's like working with DNA. And the lesson is really cool, and it's become amazing what you can do in a rather simple way. So questions, let me open it up to questions. I've taken up all my time, but if people want to hang around, I'm perfectly happy to hang around for questions. You can unmute yourself.
So isn't some of the vaccines genetic based, and does that have to do with genetic mutations, and your thoughts on that?
Yeah, it's really interesting, completely different way of doing it. So normally to develop an antibody, you want an antibody to the virus. What they might do is inject a dead virus into you, and your immune system would make the antibodies. Or you could even put a protein from the virus into your body, and your immune system would say, that's a foreign protein. What's happened with Moderna and Pfizer is, in fact, they are putting RNA as the vaccine. The RNA goes into the cell, and the RNA encodes, makes this foreign protein. So instead of injecting the foreign protein, you hijack your cells to make the foreign protein itself. Your immune system sees it as foreign, makes the antibody. So it's a completely different way of doing it, and it's partly based on the idea that the technology of making the RNA and manufacturing it is in some ways simpler and faster to do than making the protein and injecting that into your body. So that's the advantages of this new system. It is brand new, and so far it looks really good, but we don't know whether there might be other consequences. So far it looks good though.
Thank you.
Other questions? Okay, we got anybody here? All right, okay, well, so let me say, folks, that Martha Zuniga has provided a sort of summary of her talk with a glossary and an explanation of some of the terms she used, and I'm going to make that available to everybody. The recording of this lecture will be available on the OLI website. We'll probably do my food and biochemistry course again in the spring, and a year from now, in person, I hope that we will all get together up in the classroom at UCSC, and we'll have molecular biology with some other young, smart whippersnapper professors that we have just brought in the last few years. All right, so thank you much. Thank you, everybody.
Thank you.
Yes, thank you.
Thanks, Barry.
That was really interesting.
Okay.
Thank you.
Thank you. Bye-bye.
Okay.
It wasn't